All right. Yeah, I wasn't recording, so let me start that. All right. Let me go back to the screen share. Anyways, if you're really ill, you can contact me. If you're so ill you can't get an assignment, and I can change the date that that assignment is due for you. If you are ill and you want to turn in something late, let me know. And there is a late penalty, but I can allow you to add something late. Preparation for the class. This class is going to be very busy, very fast-paced. And it's fast-paced because we're meeting 4 times a week. We won't be faster than any other microbiology class, but for your learning, it'll be cramming a lot in a week. You should be ready for class, like you should at least look at the lab modules before we begin them, and look at the lecture and lab within 24 hours of our covering them. Attendance and participations is recommended. All the quizzes will be online, and when the quizzes are done, when they are due, you have 24 hours to get the quiz done, meaning the quizzes are all online because the class is all online. And when I have a quiz, you can access it at midnight, the night before it's due, and you can access it during the day up until 11:59 p.m. But the decision was made to allow you to have 24 hours to get the quiz done. And that's all you get. So make sure that in your work schedule you can get that done. And what am I trying to say here? We want it done within 24 hours to reduce the chance of cheating. All of the multiple choice quizzes are going to be timed. And so you might be able to look one question up, but if you look more than one up, you're going to use up too much time, and then you'll run out of time on finishing the quiz. So for all the quizzes, they're online, and try and stay up in your studying, and maybe do studying right before the quiz, and then take the quiz. Because you won't have time to look up more than about one question. Are there any questions about that? All right, student responsibilities, know the date the assignments are due. It'll be on Canvas, and the Canvas calendar will have them as well. And the syllabus, which I'm sort of going through now, has the major assignments and quizzes in the schedule, which is at the end of the syllabus. But note that most of the lectures and lab have a worksheet that is due the week that we are doing the lecture or the lab. And it's due on Saturday at 11:59 p.m. of the week of that lesson or lab. Now, if you note on here, let's go to week 1. Actually, week 9 isn't that bad. Let's go to week 2. You've got 2 lecture assignments that we're going to cover in week 2. Sometimes you'll have 3 lessons in a week, and then about 2 labs. So that means at the end of week 2, Saturday week 2, you'll have a minimum of 2 lecture worksheets and 2 lab worksheets. And so hopefully in the lab worksheets, you'll work on them while you're doing the labs. So those will be done. But if you're waiting till the end, you're going to have a lot to do on Saturday. Any questions about that? And of course, I don't have the due date of them because generally every lesson and every lab will have a worksheet that is due at 11:59 p.m. on Saturday of the lesson or the worksheet. All right, any question about that? Okay, somebody must have rejoined and you need to mute your mic. Where was I? I guess I'm under student responsibilities. You should keep your papers, all papers, and that until the final grade is given. You can see that in Canvas and then the online grading system. You should follow the student code of conduct, especially as it applies to academic honesty, cheating, plagiarism. And we'll talk a little bit more about that. If you need to withdraw, please do that by the due date. And if you're thinking about withdrawing, you know, I don't know how Yavapai does it in the summer, but normally you have a complete week to drop the class and then you don't have to pay. So if you are going to withdraw, try and make that decision up during week 1, and then you won't have to pay for this class. After that, you can withdraw and get a W instead of a letter grade, and follow how that procedure is done. Only you can withdraw and drop from the class. I do accept late assignments as long as I have not returned the grades for that assignment, but there's a penalty if you turn them in late. There's a 25% penalty the first day you turn it in late, so try and get it in when it's due. And then after the first day, there's a 50% penalty. I do not accept late assignments during finals week. Any question about that? All right, just briefly, if you plagiarize or cheat on something and I find out about it, you'll get a 0 on that assignment, even if it's one of the big assignments, like worth 100 points. So don't plagiarize, don't cheat. Essentially, everything should be in your own words. And if ever you don't put something in your own words, you're using 3 or more words of somebody else, even if it's your study buddy, put those words in quotes and then say where you got it from, okay? And the plagiarism tutorial talks more about that. Plagiarism is a real big deal. There was a US senator, and somebody found out that when he was, I think it was a graduate student, when he wrote up his dissertation or thesis, I don't remember what it was, he plagiarized. And this sitting senator had to resign his position because he plagiarized. So the big point is plagiarism is important, and don't do it if you're going to do that. I'm in class. I'm in class. Are you on my? Yes, quiet. Okay. All right. Etiquette for Zoom, just be respectful of other people and all that. You should, when you come into class, mute your microphone so that you won't make a noise when you're talking to someone or something like that. And that. All right, any question about that? If ever you have a question, please ask. Sorry, somebody's opening the door here. Suggested study methods. In this class, you really should go after, take notes and take notes and go over your notes within 24 hours. After 24 hours, you won't have short-term memory and you won't remember to fill in your notes. But you need to go over your notes within 24 hours. Just briefly go over them. And then, of course, you want to study before the quizzes and stuff like that. Plan on spending about 14 hours a week outside of class on this class. And you're going to be spending a large number of hours in class. I haven't totaled it in a week. All right, any questions about that? There's a bunch of other notes we can do for studying. I'll let you read over that. Grading. The plagiarism quiz, you do have to take it, and you must score 100% on it. You may take it 5 times, and if you don't get 100% on the plagiarism quiz, then you will earn, besides 0, well, whatever points you earn on the plagiarism quiz, you will end a 0 on the infectious disease project. This is a 100-point project, so make sure you do take the plagiarism quiz and get it 100%. And like I said, it's the same quiz, so the first time you take it, just take it again. I think the answers are reshuffled, so don't just go, okay, I took A last time, I'll take B this time, because the answers shuffle. There are 5 quizzes. They're worth 50 points, but you get to drop the lowest quiz, and the quizzes are on the assignment. I think the first quiz is during week 3, but I'll have to look at the schedule. So that's 4 quizzes times 50 points, or 200 points total on the quizzes. They must be taken within 24 hours of when they're due. There's going to be a worksheet in the lab and a worksheet in the lesson. There will be 1 worksheet for each lab for a total of 100 points. Actually, some of the later labs have more than 1 worksheet, or it's a very long worksheet. But anyways, there'll be a total of 100 points. In the lecture, most of them will have 1 worksheet per lesson, but some of them have 2 worksheets per lesson. And however many points we have, I'll total them at the end to be 100 points. And as I mentioned, the worksheets are always due the week that we are doing the lesson. There will be an identification of an unknown bacteria. Later in the term in the lab, I will email you an unknown number and then the assignment information. And your goal is to run microbiological tests identifying your unknown bacteria. You will be doing this, of course, totally online and virtually, but you'll be pretending as if you were running the real microbiology tests in the lab. Is there any questions about that? Have I made that clear? All right, no questions. There will be more information provided, and we'll talk a little bit more about that. There will be an infectious disease research project, 100 points, and you have to research a pathogenic organism of your choice. Normally, I say that each student has to have a different organism, and I think I'll try to do that this term as well. So you need to send in what your organism is, but because there's a very large number of students in this class and in the class linked to this class, I'll probably let at least some overlap because, well, I don't know how many diseases there are, but 40 of them will be an awful lot. And so, like I said, I'll probably let a few overlaps. There will be a cumulative final exam. It's worth 150 points. We'll talk more about that. I'll tell you when that is. July 23rd, there will be the cumulative final. I haven't made it yet. It will be online. Probably multiple choice, so that means it'll be timed. Any questions about anything here? All right, so here's the breakdown of the points. 20 points for the plagiarism quiz, 200 for the other quizzes. You get to drop one quiz. Lab assignments, various numbers for 100 points. Lecture worksheets for 100 points. The infectious disease project, that's one assignment for 100 points. Identification of the unknown for 100 points. And then the final for 150 points. So that makes a grand total of 770 points. And you will be graded on your percentage of the total 770 points. Here it is here. If you get 90% or more, you'll get an A, 80% or more, a B, etc. Yavapai does something funny. You have to get over 65% to get a D, and under 65% is an F. There will be some extra credit assignments. I gave you 2 already, mentioned them briefly, and there will be about 30 points of extra credit assignments. All right, any question about the grading and the point assignments? All right, these other things, you can read over them. If you have a question and you don't feel I did something correctly, I've got a procedure in here for writing up your written correction. Proper use of the microscope, we'll talk about that in the lab. Non-discrimination policy, go ahead and read that. Safety information for students and physicians. You're going to be an online class, so you won't be exposed to bugs, but you should know that information. Words of advice and comfort, I'll let you guys read over that. Laboratory safety rules, you're not really going to be in the lab because everything's going to be online, but you do need to know the safety protocols and you will be quizzed on them. So let me briefly go over the laboratory safety rules. Safety is one of the main skills that students normally learn in microbiology, especially if they're going into the nursing program, which many of you are, but any of the health sciences require safety, and that's what we teach in microbiology. And so even though you're taking this class online and taking the lab online, you do need to know the safety protocol. In the microbiology laboratory, safety is of paramount importance. You want to keep yourself safe, the other members in the lab safe, and then the environment safe. And so when you're doing the procedures, we teach you how to do that. Never take a shortcut because of the risk involved. And when we're working with microbiological organisms, I sort of skipped over that and told you to read it. The organisms we use in the lab are biosafety level 1, BSL1, or BSL2. BSL1 things don't give people really an infection. You might be able to get an opportunistic infection like from E. coli, but these are things that people are exposed to all the time, and they don't really make people sick unless they were to consume large numbers of them or get an injection underneath their skin or someplace like that. Then it would give them a problem. But biosafety level 1 is the lowest risk, and really all you have to do for that is clean the laboratory surfaces, the counters, before you're using it. And then at the end of the day, you also clean it with a disinfectant. Biosafety level 2 requires a little bit higher level. These are organisms that can make people sick, like Proteus mirabilis. People can get that infection, Pseudomonas aeruginosa, really bad, especially if they get it in their lungs. And then burn patients frequently get this on their skin. With these organisms, you have to wear gloves and a lab coat. which in the lab we require you to use, and you should have appropriate shoes where the toes are covered. And what else about that? You have to wipe down the surfaces with the disinfectant and wipe down the surfaces with the disinfectant afterwards. And then when you're leaving, you have to wash your hands. So everything in the lab stays in the lab. That's the point there. And things should not be brought into the lab, like organisms, unless it's approved by an instructor, and then nothing should leave the lab. And that's why we wipe down the counter, and then you take off your gloves at the end of the day, and you take off your lab coat, and they're kept in the lab. All right, any questions about that? And I guess you also wash your hands before leaving. And the purpose of that is to make sure that bugs aren't taken home with you, and then either you get them or you inoculate somebody else. And all your personal items, when you bring it into the lab, we have cubby holes, and your personal items are put in the cubby hole, like your book bag, your purses, your cell phones, your books, your pencil and papers, everything is kept in the cubby hole. And the cubby hole and the door that you come into are kind of considered outside the lab, and everything else in the lab is considered inside the lab. And so you put things in your cubby hole, and then you wash your hands, and then you get suited up. So everything you bring into the lab is left in the storage cabinet by the door, and you don't ever touch that stuff unless you're leaving the lab. And with the phone, what you'd have to do is come into the lab, and you're suited up, and you have gloves on, and you have a lab coat on. You have to take off your gloves, take off your lab coat, wash your hands, then you can grab the phone out of your books. Then you have to leave the lab, use the phone, come back into the lab, put away your phone, wash your hands, put on your gloves, put on your lab coat, and then you can go back and work in the lab. So it's usually a hassle to get a phone call that when you're in the lab. All cultures you work with in the lab are potentially pathogenic, and that's why students are required to wear gloves. You should monitor yourself for illness from getting any of those organisms. You must wear closed-toed shoes. This is to give minimum protection. Like sneakers don't give a lot of protection, but if you drop something and it hits your toes and you have open-toe shoes, your feet could be exposed to microorganisms. And so closed-toed shoes must be worn in the lab. A lab coat must be worn in the lab, and it stays in the lab. Gloves, whenever there's a splash hazard, whenever you're working with a liquid broth or some other splash hazard, like you're working with the dye that could get in your eyes, you have to wear goggles. I already mentioned you must wash your hands when you come into the lab. You must, before you work in your lab space, you must disinfect it at the start. And then when you're all done, you have to disinfect it afterwards. And then before you leave, after you take off your gloves and your lab coat, you have to wash your hands before leaving. Any question about any of that? Any accidents should be reported to the instructor. I think I'll let you read over that because you won't be in the lab. But generally, you drench the area if there's microorganisms in it with a disinfectant and let it sit for 15 minutes. Cover it with a paper towel. There it is, lots of paper towels. And that's the wick, the disinfectant throughout the bacteria. Keep your hair tied if it's long and away from Bunsen burners and backtoe incinerators. That's for heating our organisms and not the organism, the tools we use in the lab to sterilize them. No cultures can ever leave the lab. I'll let you read over that. If you have a cut, you put a Band-Aid on the cut and then put the glove over the Band-Aid. Did you guys hear that? I was pretty quiet there. Put a cut. If you have a cut, put a Band-Aid over the cut and then put your gloves over the cut. All microorganisms you're working with must be labeled completely with your name or initials, the date, the nature of the specimen, meaning what it is, like E. coli, or cultures from air, and then the medium used. For an optional one, you could have the temperature. Old specimens must be discarded properly. Agar plates are discarded in the appropriately labeled biohazard trash cans, and I will sometimes call them the autoclave bag. It's a trash can that is labeled biohazard, but it is autoclaved before it is discarded. So microorganisms are put in there, and assuming that it's plastic or paper towels or something like that, put in there, and then they're autoclaved, and then they'll put in the trash. So that kills the organisms. Nothing glass is put in the biohazard bag or trash can. The glass slides and cotton swabs are placed in the glass container disinfectant found near the rear of the lab. Pipettes, if they're glass, are put with the glass plastic and the labeled container. I mean, there's a container that says glass pipettes. Generally, we don't use those in the lab. Test tubes are put in the appropriately rack that is for test tubes, and there's one in the rear of the lab for liquids and one for solid, and you put the test tubes there. Supplies that are needed are in the front of the lab. I already talked about that. Anyways, you must follow those agreements on following the safety while you're in the lab. And I only went over those because you'll be tested on that material. You do need to know it, but in reality, we won't be in the lab. All right, any questions about any of that? Not hearing any questions. Let me stop the share here just so I can see. I forgot people, all right? All right, so at the end of the syllabus, I've got the schedule. So in week 1, we'll be discussing the syllabus, which I'm almost finished. And then we'll be starting chapter 1, the microbial world in you. And attached to chapter 1 as part of chapter 10, we'll be discussing the classification of microorganisms. Any question about that? Remember, we will start chapter 3. That looks like the 4th day, so on Thursday. But on at least Tuesday, we will be continuing chapter 1, and we might be on chapter 2 on Wednesday as well. But at least on Thursday, we should be on chapter 3. So chapter 1 will have a worksheet that's due on Saturday. And it looks like chapter 3 is only one day. So its worksheet will also be done on Saturday. Any question about that? All right. Your first quiz will be a practice quiz, which is due on Tuesday of week 2. And you don't have to take it, but it's a good idea to take it because it'll give you practice on taking an online quiz. For example, it is timed, but this quiz you can take twice. And all the other quizzes, you can only take them once. So you should take it as if you were going to take this once. But if you aren't happy with your answers there, you can take it again and get more points. And that is, if you take this quiz, I'll give you one and you get correct answers. I'll give you 1 extra credit point for taking the quiz. So that's just something. You won't see any of these questions in quiz 1. but some of the concepts may be the same. The point of this quiz isn't to be used as a study guide for taking quiz 1. It's just to get you a little bit of practice on taking a quiz. Any question about that? All right. On Friday of week 2, you have to take the first quiz. And if we keep to the schedule, the quiz will be over chapters 1 and 3, and lab 0, that's today, and lab 1, which will also be today. Any questions about quiz 1? Like I said, the quizzes must be taken within 24 hours. You can get on it midnight of Thursday beforehand, but it must be finished by 11:59 p.m. on Friday. Quiz 2 will be during week 3, and it will be also have to be taken by 11:59 p.m. on Friday. Quiz 3 during week 4, also by 11:59 p.m. on Friday. Quiz 4 during week 5, also on Friday. Quiz 5 on week 6, and it's also on Friday. Note there will be no quiz on week 7, and you'll have the final exam in week 8. Now, as I mentioned, I've got the schedule up here, and if we keep to this schedule, then I've got what the quizzes will be, the questions will be on. But if we fall behind schedule, I doubt we'll get ahead of schedule, but if we fall behind schedule, the quiz may not cover this section. Any questions about that? Another thing, this is the tentative schedule, but if you notice, I've got up here, it's a tentative schedule and it's subject to change, especially this term with COVID-19. I'm going to try and follow the schedule. If we get ahead of schedule, that's not a big problem. We can just stop early. And I have in the schedule a few catch up and review. So if we fall behind schedule, well, we can cover chapter 6 and not have catch up and review. And then on week 5, I've got a whole day for lecture, catch up, and review. And if we're on schedule, we will just review that day. Okay? If we're behind schedule, then we will have the lesson on that day. Any questions about that? I think that's pretty self-explanatory. And then I've got on July 20th, a day set aside for reviewing. And that'll be before the final on the 23rd. And how many days? Let's see. What day is the 23rd? Does anybody know? It looks like it's going to be Thursday if the 20th is Monday. And then nothing scheduled for the 21st and the 22nd. So you can study on those days. Of course, the 21st, your unknown projects is due. All right, any questions about the schedule? All right, let me say something here. Wherever I've got an assignment that we're going to cover, there's 4 of them. These are assignments that are not in the custom textbook, so you'll need to get all of these from my web page. That's antibiotics, host defenses 1, host defenses 2, and parasites. Now, I'm hoping to cover parasites, but if we fall behind schedule, I won't even cover parasites, meaning I will plan on taking the review. I'm not going to cover anything on the 20th, even if we're behind schedule. And hopefully we'll only not have that lesson. You know, that's 2 days for that, and there's 2 days for that. So we have 2 days that I can, if we really fall behind schedule, hopefully we won't be more than 2 days behind schedule. All right, any questions? If not, I've already showed you the lab web page. Let's come up here and state that, oh, I need to talk about chapter 2. Chapter 1, I've got up there a study guide. If you look at that, always before the study guide, I've got a page. Let's see if it shows. Can you guys see this? I'm not gonna blow it up. So hopefully you guys can see that. Nobody's saying no. On the very start, I've got what the objectives are for this lesson. And then after the objectives, I've got the class notes. Oh, excuse me, this is chapter 1, which also has chapter 10 in it. So that's the objectives for class chapter 10. There's the chapter 1 notes. Now you could print this out and add your notes to it, or you can just take your notes. That's up to you. But I always have this. The nice part about the notes is you'll get the lecture material in a different format. Generally, this format has complete sentences, and my lessons that we'll go through has it in abbreviated sentences. Any questions about that? Let's briefly talk about chapter 2. We're not going to cover chemistry. In theory, you've had this in your prerequisite class, but if you're weak on chemistry, I put the notes there because I'm not going to go over this material. Oops, went too far. But there are some things you should know, like the 4 major biological molecules. Those are proteins, well, carbohydrates, proteins, lipids, and nucleic acids. And I'm not going to talk about the bonds, but you should know what a covalent bond is, an ionic bond, and then hydrogen bonds. So I'll expect you to know those terms. And then you probably should also know the functional groups. I'm scrolling towards it. Functional groups give a property to a molecule. I think it's this slide, but boy, can I get it to show. There we go. The ones in red, I expect you to know, like a carboxyl group is found on a protein. We're looking at it right here, and an amino group. Anyways, if you're weak on chapter 2, I've made that lesson available. You can take it and study it on your own. Chapter 2 is chemistry. We're not going to cover chapter 2. Any questions about that? All right. Let's start chapter 1. Chapter 1 is the microbial world in you, and we're going to cover a little bit of chapter 10, classification of microorganisms. It's page, if you have the custom textbook, page 272 to 281. And I bet nobody has the custom textbook. I don't know what it is, and it's chapter 10 in the full textbook. Usually what I do is when I start a lesson, I'll give you the first slide, which will give you the major goals. That is not the only goals you need to know. They're just the major goals. If you want to know all the goals you need to know for a lesson, take a look at the objectives, and those are on the study guides. Okay, I should mention that sometimes most of the times I have just one study guide, and it'll be the objectives at the top and then the class notes in the study guide. But sometimes I've broken it out, the class objectives and the class notes. So I've broken it into two files, and hopefully that won't confuse you. Also, when I'm showing you the major goals, I give a rough outline for what we're going to do. All right. So for lesson 1, you need to know what a microbe is. You need to know what a microorganism is, a germ, a pathogenic organism, a parasitic organism, a microbiota, what an infectious disease is, what an emerging infectious disease is, what pasteurization is, and what Koch's postulates are. Any question about those terms? We will go over them. We'll also in this lesson go over a brief history of microbiology and discuss the importance of microbes. Let me go over the slideshow of this because it's kind of small. We're going to be talking about the importance of microbes. Here you're looking at the nitrogen cycle. Nitrogen can start as into nitrogen gas in the atmosphere, and that nitrogen can be fixed by bacteria and archaea into organic compounds, and those can be decomposed by bacteria, archaea, and fungi into ammonia, and ammonia can convert into ammonium ions, and both of those can be taken up through the soil by plants and algae, which algae would do it in the water, but you get the point. But they don't really like ammonium or ammonia. But ammonia can be converted into nitrite, which can be converted by bacteria into nitrate. And this is what green plants really like. If you buy a plant fertilizer, it'll have nitrates in it. And they will uptake that, and then plants will convert it into organic compound as part of the plant. And then that will be consumed by others, consumers. And that's how we get our organic nitrogen. And yes, let's say you're only eating it from beef. Well, the cow got it from eating the plants. Sorry, I lost my mouse here. And then when they die, it'll be decomposed into nitrates or into ammonia, and then the nitrogen can cycle, and the nitrate can be converted by microorganisms into nitrogen gas in the atmosphere, and then the nitrogen cycle is then complete. The point of this is how important are microbes in this cycle. And here you're looking at the steps where a microorganism converted the nitrogen in one part, wherever it's red is the step, a microorganism converted it from one form to another. And as you notice, almost all of the steps of the nitrogen cycle are happening because of a microorganism, meaning microbes are crucial to the functioning of the nitrogen cycle. Any question about that? Well, here's the point. In one place I said microorganism, in another I said microbes, and nobody asked about that. So is that clear to you that a microorganism is a microbe and a microbe is a microorganism? They're the same terms for the same concept. Okay? Everybody got that? All right, let's end the show here, and hopefully we'll start there. Good. Biogeochemistry, rock-eating microbes. One wouldn't think that when we're looking at Mount Erebus, and I don't know how you say that in America, or rock in an ice cave, that the converting of rock breaking down into soil, that this is all geology or oceanography or whatever windology is. I don't know what that is. Biogeochemistry. And that the breakdown of rocks, everyone probably knows, makes the formation of soil. But actually, bacteria are important in the making of soil. There are microbes that break down the rocks and help form soil. Now, obviously, wind and erosion are also breaking down the rocks, and then water running. They're also breaking down the rock and making soil. But a significant portion of the soil made is made by microorganisms. So once again, on Earth, this is showing you that microorganisms are important for the planet Earth. Microorganisms help convert rocks into soil. When we're looking at the Earth's atmosphere, when we look at the oxygen concentration, I'll blow this up because it's kind of small. This is the concentration of oxygen in the Earth's atmosphere over time. And currently we're at about 21% oxygen. And as you see, this oxygen level has been increasing over time. And when you get before 2.7 billion years ago, there was no oxygen in the Earth's atmosphere. So what made oxygen right here? What brought oxygen in the Earth's atmosphere? It wasn't green plants because they didn't evolve until, where is it? I don't know. Someplace around here, okay? The first oxygen in the Earth's atmosphere happened by microorganisms such as cyanobacteria releasing oxygen in photosynthesis. And that brought oxygen up into the Earth's atmosphere to around 5% of today's oxygen. And then green plants evolved, and that allowed green plants to evolve. And once green plants evolved, then the oxygen came up to 21%. But about half of the oxygen in the Earth's atmosphere today is from microorganisms. The other half is from plants, green plants, and large plankton, like the seaweed that washes up on the Washington coast. All right, so once again, oxygen is important to the Earth. It is not oxygen, obviously important to the Earth. Microorganisms are important to the Earth. They are responsible for half of the Earth's oxygen, and they were responsible for first bringing oxygen into the Earth's atmosphere, beginning about 2.7 billion years ago. All right, any question about that? Here we're looking at a sewage treatment plant. And humans are responsible for generating this waste. It happens when you flush your toilets and wash down your drains in the sink. And they're responsible for breaking it down, right? Anyone disagree? I mean, obviously we've built all of this stuff that you're seeing here to break down the waste, right? In reality, microorganisms break down the waste. And we built the structure and we generated the waste, that's true, and we collected in this place, but it's the microorganisms in this facility and the rest of it that break down the waste. Humans don't break down the waste. Any question about that? So without microorganisms, we would be in trouble because every time you flush your toilet, there would be this waste that would just pile up. And then what would we do with it? Unfortunately, microorganisms break it down. All right, any question about any of that? The point was, I'm just trying to go over some of the ways that microorganisms are important in the Earth's ecosystem. All right. What are microorganisms? There are organisms that are too small to be seen by the unaided eye. Generally, they're single-celled organisms. They can take on many shapes, and they have different sizes. It includes bacteria, archaea, also called archaebacteria, yeast, molds, protozoans, some algae, almost all of the algae which are single-celled. It doesn't include the algae, which like the kelp that wash up on the Oregon and Washington beaches that you used to swing them above my head when I was a boy, those things. But it does include the single-celled algae. And then when we're talking about microorganisms and microbes, we also mean viruses and prions. These last two, viruses and prions, are special because they are not cellular. They don't have cells, and they're not organisms. You could argue whether they're alive or not. A prion, nobody says they're alive, but we're not going to get into that argument. They're just special microorganisms because in reality, they're not cellular, so therefore they're not an organism. And we will talk about these in microbiology. But that's what we mean when we say microbe or microorganism. They are almost everywhere, so we say they are ubiquitous. The ubiquity of microorganism means that they are essentially everywhere. They play beneficial roles in numerous processes, and I went over a few of them earlier. They are numerically the most abundant life form on the planet Earth, and we're going to study them. Any questions about that? All right, I did use the term cell, so let's go over what the heck a cell is. A cell is the basic structural and functional unit of all organisms. And obviously we're talking about cellular organisms. They're highly organized compartment. They are bounded by a thin and flexible membrane, and that is called the plasma membrane or the cell membrane. Plasma membrane and cell membrane are used interchangeably. All cells contain three components. One I've already mentioned, and that's the plasma membrane. They also contain cytoplasm, and they contain DNA. So all cells have DNA for their genome. Any question about what a cell is or a microorganism?
I don't have a question about the cells.
Hold it, two people are talking, one person at a time.
I think we're gonna ask the same question. If you can keep it full screen, because it's hard to see.
Well, there's not much to see here, so that's why I went down. Generally, when it's hard to see and there's something that you should see, I will make it full screen. Okay.
I think our issue is that we can't even see like what's written on there. It's very blurry.
You can't see that right there?
I mean, it's hard to see. It's very blurry. That is probably this like an inch and a half across on my screen, and I'm using a laptop.
Okay, yeah, I didn't realize that's small, but I can read it, and I have poor eyesight, and most of you have much better eyesight than I do. So, all right.
Well, on the
like
Zoom meeting, it has your video next to it, and then the group chat next to that, so it's even smaller.
I see. All right, I will keep it full screen whenever necessary. Like I said, I told you everything here. The cell contains three components, which you can't see that here. The plasma membrane, the cytoplasm, and the DNA. And I mentioned that cells are the basic structural and functional unit of all organisms. Okay, but yeah, if you can't see something and you want me to see it, I will put it up there. On this one, there's nothing you need to see except for the pictures here, and hopefully you can see it. What I'm asking you for here is, from this slide, where are microorganisms? And since that is most of the places, I'll ask you, Is there any place on this slide where you don't see microorganisms? Like, are they not found in the water or on the soil or the rocks, whatever that is, or on the plant life? Nobody's answering. Okay, do you not find microorganisms in the ocean or in the air? I sort of went to the next slide. I didn't mean to do that. You do find them in the ocean and in the air. You find microorganisms up, on average, about a mile in the air. And that is especially spores sort of drift with the wind, and they can go up a mile in the air. You find microorganisms in the soil. You have to go down more than a mile, so you can find microorganisms down in the soil about a mile. Okay? You find him, of course, on the young man and on his pet everywhere. So every inch of this and every inch of that, you find microorganism. You find it in every inch in the desert as well as in the air. You find it in the rock or the snow. You find it everywhere except this is a volcanic vent. You can sort of see lava there, the red. And that's the only place you would not find microorganisms. Any question about that? So you find microorganisms almost everywhere. And where is everywhere? Environments where microorganisms are found are the land and the water. You find them in the atmosphere up about a mile, subsurface under the land or under the ocean, and this is the ocean floor. You can find them about 1 mile underneath the ocean floor or 1 mile underneath the top of the land. You find microorganisms on plants or animals. You find microorganisms generally wherever liquid water is present. Microorganisms will be growing and will be present. And you find them in temperatures down to a minus 20 degrees Celsius. I don't know what that is. Oh, there it is. Minus 4 degrees Fahrenheit. And up to 120 degrees Celsius. That's 250 degrees Fahrenheit. This is temperature that you might have cooked something in your oven. Not very many things cook at 250, but some things cook there. Like I cooked, oh, what the heck is that? Nuts or I forgot what that's called. Tricks, snacks at that temperature. Some cookies, but not very many cookies cook at 250. All right, I think this is a good place to stop. And we're at a stopping point. So I'll stop here, and then at 2:30, we will continue with the lab. Are there any questions? All right, if there's no questions, I'll say goodbye, and I'll see you at the lab at 2:30. Okay?